All right. So just everyone, yeah, everyone is turned off the microphone. Thanks.
Thank you very much. Well, thank you very much for the opportunity to present my work. So I'll first start by describing our experimental setup and then present some results we published a couple of years ago, but then I will end with some newer unpublished results. Yes, so I'm a scientist based at the Inselspital here in Bern. And so I'm going to try to introduce you so broadly to the microbiota. So I'm sure you all know about the microbiota. Microbes are present on all external surfaces of our body, and this happens from birth. And I'll come back to that particular point later on. Most of the microbial diversity is contained within the intestines, but commensals can occasionally reach the systemic compartment even in healthy conditions. For example, if you have microlesions in the mouth or some small wounds. And then I think over the last 10 years, a lot of work has been done on studying the microbiota, and several studies have really shown that the diet is a major, if not the major modulator of microbial composition. And so in the context of humans, obviously, we are all unique from our genetics. We are all unique from our microbiota composition, and we are fairly unique in terms of our diet. So this composition then is quite tricky to study. Another piece of introduction here, how we accommodate such diversity of bacteria in the gut. So here I'm showing the intestinal immune system, and mostly under the epithelial cell layer, we have the lamina propria, which contains a lot of immune cells, which samples the antigens from the gut lumen. And then they stimulate B cells, which can expand and form germinal centers, mostly into two compartments, the Peyer's patches and the draining mesenteric lymph nodes that are further away. And those two sites are the main sampling sites that we use when we look at the mucosal immune systems, and I will show that later on. Yeah, so the microbiota is very, very diverse, and I just here put some numbers for people to have an idea. And we do have obviously a very diverse B-cell repertoire. But it is a very interesting and challenging problem for the immune system to accommodate this diversity and the antigens represented in here for the B-cell repertoire, and how much of a footprint the system can have on the repertoire or not. And then obviously this exposure to the microbiota happens very early on at birth. This establishes a pre-immune repertoire, which then therefore influences immune responses to pathogens later on, and also affects susceptibility to autoimmune diseases. And I'm sure there are some connections to the previous talk as well in terms of this microbiota crosstalk in type 1 diabetes. We are not the first ones who are trying to understand the forces who shape this antibody repertoire. So here it's just looking at a study from Victor's group, Victor's a few years ago, where they tried to dissect the contribution of the genetic background or stochastic private responses and the antigen exposure to different B-cell repertoire. And one aspect that they had shown then was that the private responses tend to shape a large part of the repertoire size and diversity in antibody secreting cells. And while in humans, obviously, as I mentioned earlier, these responses appear to be private, it's because then we have an individual microbiota and we tend to follow a specific diet. So our aim was to try to see if we can standardize the diet and the microbiota. and try to dissect these and therefore provide more of public responses for us to study those systems. So how can we study microbiota effects? First of all, we need to standardize the host genetics and the microbial composition. So we minimize the individuality and then maximize the public response. And therefore, then the first questions we really wanted to address was if the route of exposure, if we provide these antigens mucosally or systemically, influences the B-cell repertoire. And in a second time, then how does this baseline repertoire shaped by the microbiota respond to pathogens? So in order to do that, we used inbred germ-free mice, which are maintained in very stringent conditions and are devoid of any pathogens and microbes in general. And we can then colonize these with specific defined microbiota or even single microbes. We used also a tool that was now established quite a while ago in our lab of a auxotrophic strain of E. coli, which is deficient in the synthesis of the cell wall, which, so in vitro, if we provide the precursor in the medium, then the bacterium grows fine, but then in the intestine, then this bacterium replicates a little bit, and in 24 hours, then the bacteria dies and the mouse becomes germ-free again. So this experimental tool now allows us to uncouple microbial exposure from permanent colonization, and therefore better control the timing, the dose, the exposure route, and therefore also look at sequential exposures to different taxa. We have now also further expanded the range of auxotrophic strains we have to other taxa. So the first experimental design was to try to provide a selective mucosal priming and a selective systemic priming. So delivering then either orally or IV defined doses of the microbe. And we tested different strains from very low, different dose, from very low dose to more physiological, meaningful doses. But we also made sure that, especially IV, we delivered doses that never triggered conditions like sepsis, and the mice remained healthy. And therefore, after 21 days, then we analyzed the B-cell repertoire. So this is bulk repertoire here. And then we analyzed the repertoire for several organs. Here I'm showing IgA, but we also looked at other isotypes. And we also broadly saw this compartmentalization. So if we look now, the repertoire in this MDS plot, we obtained a distinct repertoire, whether we provided the same pool of antigens mucosally or systemically compared to the germ-free. And as you can see here, we also, by this standardization, we also increased the overlap, and we can see that we have a higher clonotype overlap within each group. And then on the right, we can see the top 100 shared clonotypes ranked by their clonal abundance, and we can see that we identify some patterns specific to each group. So the first message is that we can then see a distinct repertoire raised against the same pool of antigens according to the priming route. We then carried on with the same experimental design, but this time we changed the readout and we looked at antibody binding to bacteria by flow cytometry. And so following here systemic priming, we can see that in the serum IgG, we mount a robust response. And conversely, after the mucosal priming, we see in the intestinal fluid that we mount a proper IgA response. What was interesting to see also is that after systemic priming, we do mount a small response in the intestinal fluid, whereas after mucosal priming, we have a bit also of serum IgG response. So we have a compartmentalized response, but there is, to some extent, a cross-compartmentalized effect as well. Now we went a little bit further, and now we used single cell a repertoire analysis where we could obtain then heavy and light paired chains. And using those, we could build a network using Levenshtein distance. And we could look at a couple of facts. First of all, the connectivity in both the MLN IgA or spleen IgG was most of the connectivity was carried by the heavy chain. That was in confirmation of what others have shown as well. And then what was interesting in the case of the mucosal IgA response is that a significant portion of the connectivity connected clonotypes that were found in the germ-free to the gavaged condition. Meaning that we trying to build new responses, expand responses, sorry, that were already found in germ-free. Whereas when we look in the spleen after IV exposure, most of these expanded clusters seems to be rising from clonotypes that were mostly seen in the IV group. Right. So in the second time, then we asked what happens if we come with a subsequent challenge after an initial exposure. So we have a few experimental groups here. So in purple, we have first an E. coli exposure. In blue, we have a Salmonella with another reversible strain exposure. And then we have in orange and red, we have first either an E. coli and Salmonella challenge, followed by the converse strain challenge. And so here, the readouts we can see in the specific antibacterial IgA response in the intestinal wash. And so if we look at the E. coli specific response, we see that after priming with E. coli, we mount a robust response. But interestingly, if we prime with E. coli and then follow up with a Salmonella challenge, we tend to dampen this response. And we, to some extent, observe the similar behavior when we look at Salmonella specific response. We have an initial response to the Salmonella challenge. And then when we follow that challenge with a E. coli secondary challenge, we also tend to dampen and contract the IgA response. Here, this dendrogram shows a very condensed view of the repertoire as another readout. And here it was interesting to see that the repertoire specific after the E. coli response and the challenge where the second challenge was E. coli, they tend to cluster together. And similarly, the repertoire from the Salmonella challenge tend to cluster with the repertoire where the Salmonella challenge was the second one. So the repertoires tend to be dominated by the latest challenged exposed. Yes, and that makes sense in the context of a fairly limited in diversity IgA repertoire, as it's been shown by others before. Now, if we look in a similar design, but for the systemic IgG response, now you can observe that whether we look at the E. coli specific response after an E. coli challenge or the E. coli challenge followed by Salmonella, the robustness of the response is comparable. There is no dampening in this context. And it's similar for the Salmonella specific response. And if we look at the repertoire, we can see that we tend to generate in every group a distinct repertoire from the other group, meaning that the repertoire is here, the IgG repertoire is more flexible and can accommodate wider diversity and more unique responses. So summary so far, we have seen that we can mount a distinct repertoire that can be shaped by the exposure to the same set of antigens, either provided mucosally or systemically. I didn't show here, but it's in the paper that we also tested the thresholds, so the amount, the dose that we need to provide, and the thresholds are higher mucosally than systemically. And it's mostly the late stage of B-cell ontogeny that are shaped by the exposure to the microbial antigens, and that we have a limited expansion of IgA, but not of IgG as a functional response to a secondary challenge. So far then we looked at the microbial effects, and then we were wondering how the diet could modulate these responses as well, and especially in the case of IgA. The background for this is that IgA secreted plasma cells in the intestines are highly mutated. It's been shown in both humans and mouse. And we also know that the germ-free mice, also the IgA is also heavily mutated. So we therefore looked in germ-free mice born and raised on either the standard sterile chow that is used in the germ-free facility, or chemically defined isocaloric diets where the sources of calorie is either starch or fat. And we can see here for these mice that the percentage of IgA plasma cells in the lamina propria is reduced in both chemically defined diet compared to the chow. The serum IgA concentration to some extent is reduced in those two diets. And then the follicular T helper cells and the germinal center B cells in the MLN were also reduced. We therefore wondered what was the impact on the IgA repertoire in this compartment. So we sequenced this, and we could see that in the MLN, we can distinguish the chow repertoire from the chemically defined diet repertoires. And here again on this top 100 shared heat map, we can see that we have some specificities. It's more loosely, but some clonotypes seems to be more present in the chow compared to maybe in the chemically defined diets. And in this column, we highlight the which clonotypes are mutated. And it seems that most of the mutated clonotypes tend to be found in the chow and not in the diets. And another way to look at this is if we build networks, and then we rank the clusters in this network by their degree distribution. So the higher the degree, the higher the connectivity between the clonotypes. And so we tend to lose the more connected clusters that we can see in the chow in the chemically defined diet, highlighting that we have possibly changed the structure and the composition with the diet in this context. So to try to dissect and understand a little bit better what's going on under here, we performed some metabolomic studies, and then we identified that the LPS concentration was heavily reduced in the chemically defined diets compared to the chow. So the LPS pathways are very well characterized, and they tend to depend on the mediated TRIF and TLR4 signaling pathways. So we looked in mediated TRIF germ-free mice, knockout mice, and we could see that the germinal center B cells were reduced in those mice. And then if we look within the mediated TRIF knockout background at the effect of the diet, we could see that again, the number of germinal center B cells in these tend to be more comparable. We didn't have the TLR4 knockout in germ-free mice. We had it in SPF background, but we could even see in this background that the germinal center B cell also were reduced in this background. So we think that the main difference is that the elevated dietary LPS levels in chow contribute to germinal center activation in a mediated TRIF and TLR4 signaling dependent way. And so it happens, therefore, independently of the microbiota. And so as an outlook for this presentation, then we've seen that the microbiota composition affects both the systemic and the mucosal repertoire. I invite you, if you want to look more, we've also explored the IgA functionality, and that was published in this paper from my colleague. We also then showed that the diet already impacts the responsiveness of the IgA repertoire in absence of the microbiota. And we are therefore continuing to look into this in several ways. First, we think that our model is a good model for what happens in early life and in the neonatal window, also around and before and after the weaning reaction. So we are now trying to identify specificities for antibodies that are produced before and after weaning, which consist in a major shift in the diet. And with this, I would like to thank Professor McPherson for allowing me to work on these projects in the lab, and then all my colleagues who have performed the experiments, and our collaborator, Victor Greif. And thank you for your attention.
Oh, sorry, maybe you were not hearing me. Yes, sorry. I don't know if there is any questions, but I have one already. So in one of your analysis, you showed that there is a contraction of the repertoire,
Yes.
right?
Yes.
And it was towards the last, let's say, antigen that was provided through the challenge.
Yeah.
So you look at this right after the challenge, right? Or a couple of weeks after?
Yes, yes. We look at this 1 week after.
1 week after. So what about long-term repertoire? Because you might, 1 week after, you might just being looking what happened after a strong immune response to a given
Yes.
antigen. So maybe at a later time point, you will find back this balance between the different triggers or antigens at least. And the second question that is related, it's whether you looked at the memory cells and whether this is enriched, those specificities or the response of these BCRs in the memory phenotypes.
So the first part, so in this clean defined system with very few perturbations, actually the differences can last for quite a long time.
Okay.
We've seen them last up to half a year.
Okay.
So that's obviously because we are in a very undisturbed system. We've looked at memory B cells in the bone marrow, and we could see also the difference, but we didn't see a particular enrichment in that context. Compared to other
subsets. Okay.
Yeah, subsets.
Thanks, Alex.
Yeah, I had a question about the germ-free mice and the mice that were kind of seeded with specific pathogens. So you were showing that the systemic response, the IgA, I guess, levels seem to be not dampened when you have the secondary challenge, but only when you have the challenge happening through mucosal exposure.
Yeah.
I was wondering if you think this could be due to the fact that mucosal exposure is specifically targeting a limited set of GI-based lymph nodes. And I think that those lymph nodes tend to have pretty restricted T cell repertoires, as far as I can tell. I don't know why that is, but perhaps the T follicular helper cell repertoire is more restricted, and then that kind of causes I don't know, the germinal centers to have to compete within the same areas as opposed to a systemic response where you can have, all kinds of different lymph nodes elicited and then you don't have direct competition within the same lymph node, something like that. Yeah.
Sure. Yes, it's very true. I mean, both the TCR and the BCR repertoire are constrained in the gut. So the amount of diversity that is available, us and others have shown that even if you do multiple challenges and if you look in humans, the repertoire in IgA in the gut tends to not increase. So that also explains when you come with subsequent challenges, you replace part of the repertoire, but you do not expand. And that probably comes because these cell numbers are limited. I know that some people were trying to, and it's a very difficult experimentally, to try to look at individual pairs patches and see if you can distinguish complementary patterns and things. But it's, but I think it goes in your direction. Yeah.
And then I just had another question. So if you're looking at I guess the effect of diet on the repertoire in the gut. So there are some really interesting autoimmune models that can be induced through diet-based changes in the gut. For example, there's this collagen-induced arthritis model in mice, where if you give the mice diets that are high in indole, that can induce collagen-based arthritis. Whereas if you deplete indole from the diet, they don't get the autoimmune disease. So, but no one has really looked at the BCR repertoire in that context. So I'd be kind of interested to see if you could expand this into GI-based autoimmune dysregulations.
Yes. I mean, there are definitely some interactions there. What was our concern is that via the diet, you can obviously impact directly the repertoire, but then you can modulate the microbiota, which then can also impact the repertoire. So we were at this level trying to disentangle the two. But for example, and that was what I was thinking after the talk of Aaron before, is that we do have a lot of B cell clones which respond specifically to flagellin, for example, which also bind to insulin. So there is a degree of interaction there of expanded clones that respond to antigens from the diet, from the microbiota, and then they might be as a background level from which that can expand or respond to subsequent changes and then end up with autoimmune diseases. So we're trying to, with our reductive system, try to disentangle the contributions from these different sources to try to hopefully understand this later on.
Great, thank you.
Thank you.
Felix?
Oh, hi. Just to throw in a different complication, I mean, we know that the immunoglobulin genes of different strains of mice can be entirely different, and it'd be a lot of work, but if you did it with a couple of different strains, you'd be able to manipulate the genetic component too. So I wonder if you were thinking about that or...
Not specifically, no, but it will be very relevant indeed. Yes, to look at that as well. Yeah, sure.
Great. I don't know if there is additional questions. No, I don't think, apparently not. And we are perfectly on time. So thank you very much, Aaron and Julien, for your great talk. Thank you to the audience for the very nice discussion. Just to remind you, the next AIRR-C seminar series will be on January 25th, and we'll have Enkelija, Miho, and Stefan Shadgen that will be presenting. And also for everyone, so this seminar has been recorded, so it will be posted on YouTube, on the YouTube channel of the AIRR community. Thank you very much, and have
the
a
Porto
good
meeting.
day.
Mention the Porto
and the
meeting.
Porto meeting also. Thank you, Felix. So we'll have the AIRR community annual or biannual meeting next year in Porto, Portugal, at the University of Porto. So you will be informed by beginning of January for the registration, but the information is already online on the website. So you can check and save the dates for the meeting, June 3 to 6.
Thank you. Great.
Thank you.
Bye.
Thank you.